I'm going to make Gerald wait one moment longer while I take this time to thank everybody in the audience for coming early and staying late. So you all deserve our applause. Thank you for that. And this will also be the last time that I may see all the judges in one beautiful line. And so you have my heartfelt thanks and the thanks of everybody in this room for all the time and effort that you have put into all the analysis, discussion, and decision making. So thank you. Okay, now, this is our last individual presentation, and it's in the area of biochemistry. Gerald Meixiong is from Lakeside High School in Evans, Georgia. His mentor is Dr. Quansheng, professor of neurology at Georgia Regents University. Gerald.
Thank you. Hi, good afternoon. My name is Gerald Meixiong, and the title of my talk today is Cell Cycle Regulated Membrane Association of NuMA, a novel pathway for efficient chromosome segregation. Cell division is the process by which a parent cell divides into two daughter cells, and it is important for the development of life. For example, upon fertilization, a human egg begins a period of rapid division, and in one day, it's divided into 2 cells. In 2 days, it's divided into 4 cells, and it continually divides, and in only 9 months, a fully functioning baby is born. Cell division is also very important in adults for tissue regeneration and repair. For example, upon skin damage, epithelial cells must continuously divide to ensure for proper wound healing. Uncontrolled cell division, however, causes cancer, so cell division must be properly regulated. Cell division is regulated by the cell cycle. The cell cycle consists of 2 stages, interphase and mitosis. During interphase, the cell grows and DNA, the genetic material, is replicated. During prophase, the first stage of mitosis, the nucleus breaks down and DNA is condensed into chromosomes. These chromosomes then align along the equator of the cell and are pulled apart. During telophase, the cleavage probe forms to separate the 2 daughter cells. I'd like to emphasize that during anaphase, chromosome separation is very important to ensure that each daughter cell obtains a copy of the parent DNA. How are chromosomes separated? There are many different forces which act on chromosomes for their separation, and one of these has been proposed to be dynein at the cell cortex, or the inner layer of the cell membrane. The orange structures shown here are called centrosomes, with the blue structures coming out of them called microtubules. Here is an image of a cell in anaphase with this region enlarged with the centrosome, cell cortex, and microtubule, with the minus end of the microtubule attached towards the centrosome. Dynein is a minus end directed motor protein which attempts to move towards the centrosome. However, if it is anchored, it cannot produce movement and in turn pulls the minus end of the microtubule towards itself. In a classic model, dynein is anchored at the cell cortex by the protein ternary complex of NuMA, LGN, and G-alpha. In this complex, G-alpha associates with the membrane and binds to LGN. LGN is an adapter protein and can bind to NuMA. NuMA then associates with dynein and recruits it to the cell cortex. This pathway is active during metaphase and anaphase, and evidently, in this pathway, NuMA is a very important protein because it most directly associates with dynein. Now, what is NuMA? NuMA stands for the nuclear mitotic apparatus protein and is mainly nuclear during interphase, although its nuclear function is still unknown. It can associate with microtubules and has functions in spindle organization and assembly. If NuMA is depleted in cells, cells would exhibit mitotic arrest and eventually die. In the ternary complex, NuMA associates with dynein and LGN. To see how NuMA can recruit dynein to the cell cortex, I created a construct with deleted microtubule binding site and nuclear localization signal that would not associate with microtubules or be localized to the nucleus to eliminate any confounding factors. I transfected this construct into cells which are also expressed with Venus, a fluorescent protein which can be observed under a fluorescent microscope. In cells, Venus alone did not show strong membrane binding. However, the double deletion mutant of NuMA showed stronger membrane binding. To see if this membrane binding was due to LGN, I created a separate construct, NuMA 1981 to 2060, which is downstream of the LGN binding domain. Surprisingly, this construct also showed strong membrane binding, which shows that NuMA can actually bind to the membrane independently of LGN and thus G-alpha. However, upon the deletion of the final 20 amino acids of this region, the membrane binding was lost, which shows that the region 2041 to 2060 is required for the membrane binding of NuMA. However, this experiment does not show if NuMA can bind directly to the cell membrane or whether this binding is mediated through another protein. However, upon analyzation of the amino acids of this sequence, one can find many positively charged lysine and arginine amino acids, and these may be able to bind to negatively charged membrane lipids. To test this hypothesis, I conducted lipid binding assay in which I transformed a PGX vector encoding for GST NuMA DNA into bacteria, purified the protein from the bacteria, and incubated it with lipids dotted on a nitrocellulose filter. If NuMA can directly bind to the cell membrane, then the fusion protein should be able to bind to assayed lipids. GST alone should not be able to bind, and membrane binding deficient NuMA also shouldn't be able to bind. Bound GST would be detected using antibodies against GST and then visualized using secondary antibodies. Unbound proteins and antibodies would then be washed away. Here's the experiment with 15 different membrane lipids dotted on a nitrocellulose filter. As expected, GST alone did not show binding to any of the assayed lipids, and membrane binding deficient NuMA also couldn't bind to any of the assayed lipids. However, this region of NuMA, 1981 to 2060, which was shown to bind to the membrane, showed significant binding to membrane lipids such as phosphatidic acid, PIPs, cardiolipin, and sulfatide. This shows that NuMA can actually associate with the membrane through direct binding to membrane lipids. To see if NuMA can bind to PIPs in vivo, I sought to deplete the amount of PIP2 in the cell membrane using ionomycin, which is an ionophore that increases the intracellular calcium concentration and activates phospholipase C, which catalyzes the breakdown of PIP2, thus depleting the amount of PIP2 in the cell membrane. Here are transfection experiments showing that this region of NuMA can bind to the membrane, and this region, 1981 to 2040, cannot. Compared with untreated cells, cells treated with ionomycin showed significantly reduced membrane binding. Here's the data quantified in this table. This shows that NuMA can associate with the membrane through direct binding to PIP2, with depletion of PIP2 in turn reducing the amount of NuMA membrane binding. I've shown that NuMA can associate with the cell membrane through a region in its C-terminus 1981 to 2060. However, upon transfection of this fragment in mitotic cells, the membrane binding is not significant until the anaphase stage of the cell cycle. This suggests that the membrane binding may be cell cycle regulated. Interestingly, NuMA has a CDK1 phosphorylation site on its membrane binding domain, threonine 2041. CDK1 is a serine threonine kinase which regulates the cell cycle, and its activity is inhibited upon anaphase due to cyclin B degradation. To see if CDK1 phosphorylation affects the membrane binding of NuMA, I created two constructs, T2041A, in which I mutated threonine into alanine, an uncharged amino acid which would prevent phosphorylation, and T2041E, in which I mutated threonine into glutamic acid, a negatively charged amino acid which would mimic the effects of phosphorylation. The T2041A construct showed strong membrane binding throughout both prophase and anaphase, suggesting that the membrane binding of NuMA may be negatively regulated by CDK1 phosphorylation. Complementing this result, the T2041E construct, which mimics phosphorylation, did not show membrane binding at any stage of mitosis, similarly to the membrane binding deficient region of NuMA. This shows that the membrane binding of NuMA is cell cycle regulated and likely by CDK1 phosphorylation. To see if the membrane binding of NuMA can affect the chromosome separation of cells, I used time-lapse imaging to measure the distance between chromosomes at different times during anaphase. I expressed membrane binding deficient NuMA 1 to 2040 and full-length NuMA in NuMA depleted cells. Cells expressing membrane binding deficient NuMA, shown in red, actually showed slower chromosome separation when compared to MDCK control cells and cells expressing full-length NuMA. This shows that the membrane binding region of NuMA is actually required for efficient chromosome segregation, and cells which express membrane binding deficient NuMA show slower chromosome segregation. In summary, I've shown that NuMA can associate with the cell membrane independently of LGN and thus G-alpha. It has a membrane binding region which can directly bind to membrane lipids such as PIP2, with depletion of PIP2 resulting in decreased membrane binding of NuMA. This membrane binding is also cell cycle regulated and likely by CDK1 phosphorylation. The membrane binding is also required for efficient chromosome segregation, and cells that express membrane binding deficient NuMA exhibit slower chromosome separation during anaphase. To conclude, I propose a working model where NuMA can directly recruit dynein to the cell cortex and aid in chromosome separation. This model is active during anaphase only, whereas the previously mentioned ternary complex is active during both metaphase and anaphase. Both models recruit dynein to the cell cortex and aid in producing robust pulling forces for chromosome separation. This new pathway may act as a druggable target, and if drugs can be found which promote this complex, then it would be good for wound healing, and if drugs can be found which inhibit this binding, then it would be good for cancers. I'd like to thank Dr. Quansheng Du of Georgia Regents University for being my mentor and allowing me to work in his lab. I'd also like to thank Dr. Zhen Zhang and Xingwen Wan for helping me with technical supervision. And finally, I'd like to thank the Siemens Foundation, College Board, and George Washington University for allowing me to present my research. Thank you.